this should be familiar.
There you go.
You want to do polypeptide data on the right, or not polypeptide. Wait, where is it though? G model product, polypeptide data. But instead, you want to click the link for GenBank. That's what you want to do.
GenBank.
So, yeah. You want to go to the right, GenBank.
Can you move her back up?
And then once you're on this page, you should be able to hit run. Button.
Am I in it? No, I don't want to be in it.
right there.
Sometimes it takes a while.
Am I in that video?
Oh, this one's
No,
done.
no.
But, sometimes it takes a while. You might want to refresh the page if it takes more than a minute or
Big
two.
circle.
And then once it's on this page, all you really have to do is change this to Homo sapiens. Organism is Homo sapiens. So all are good. And then just hit blast. And that's it. And you go wait. Okay.
Wait, there's a lot of Homo sapiens.
Yeah, but they're all 9606. Just make sure it's 9606.
Okay.
So they're all the same. Yeah. It's just all spelled wrong. So black, you guys did, what human homolog did you guys do? CSAT? So human CSAT is complex with PLP, CSAT isoform 1, so there you go. Just use isoform 1, I guess. And that's it. Right?
Wait, what is oh, no.
Yeah. So pretty much, if you look, the top hits are CSAT protein in Homo sapiens, which is expected because you've already looked this up before. But this is evidence that's actually showing that it's a homolog of CSAT, right? And for vestigial, it'd be VGL. Vestigial would be VGL 1 and 2. Cinched, ebony. Probably some enzyme, right? Which one did you guys talk about?
Isn't that just the first one?
Yeah, so it came up. Cinched?
Yeah.
Baskin?
Yeah.
FSCN1?
Yeah. Two.
Two, or whichever one you wrote about, yeah.
Yeah.
I mean,
There's only one. There's only one.
No, there's the first one
Yeah,
just says
whichever one you talk
acting,
about
but
in
it
your
doesn't
paper,
really
use that
relate it to anyone.
No,
And
use
the
the one that you
third
talked
one,
about in
it
your
was
paper.
something related to sperm size,
Yeah.
which
Yep. Once you
doesn't
click on
really
it, this
affect
comes up.
anything.
So I'll quickly explain how to interpret this. So actually, I'll quiz y'all. So if you see a plus here in the middle, so the top is your fly, the subject at the bottom is your human. What does anybody want to take a guess what the plus means?
Insertion.
Pretty close to like what it is. Right?
That's
It's like
correct.
it's like one off or something like that.
No, so the plus means it's similar.
Yeah,
So it's either hydrophobic, hydrophilic, they're small, they're huge, they're charged, right? They're similar. And that's what the plus means. And those are your positives. So positives means similar in function.
Explain that in the video. Okay.
And if you see an amino acid in the middle, that means a direct alignment. So for example, if you see an A that matches up, or a P that matches up, or an R like arginine, or whatever, that means they're exactly the same. And then the blanks mean that they're nothing similar, right? So whichever one you find, pretty much just present this as a figure. Like screenshot it. You guys know how to do this. We've been through this before, but you know, just screenshot this. You know, put it in PowerPoint or whatever. And make sure you explain the positives. Make sure you explain the identities. If there's an e-value, yeah, the e-value is 0, right? And the score. So just explain all these little things in your description. But this is your protein blast. Not too hard, right? So positives are the pluses, which means similar.
But how do I
So arginine and K is light, right? I see. Okay. So those are both what? Positively charged, right? Okay. Yeah.
MChem.
Don't want to get called out on that.
No one
yeah, so it just means that they're similar. So present this, talk about the numbers that you see, the different positives, the different, you know, whatever you want to talk about. But this will be one of your figures, and that's why I want. Okay.
Wait.
You want all of them?
Yeah, just screenshot the whole thing. In fact, you probably want the name too, because it shows that it's CSAT, right? So
Wait.
just go from like here all the way down to here.
So we have to just talk about the percentages that are up there, the positives.
Yeah, all the numbers up there, and then talk about how it's a homolog of CSAT.
Homolog of
And that's,
CSAT.
you can tie that into your introduction, right? Or not, yeah, your introduction, because you talk about
FS
CSAT.
and
Homolog
C2.
of FS.
Or for you guys,
FS
FAS.
FASN.
yes.
And then you want us to explain all the way
Two.
Oh yeah, well, obviously you'll have the figure legend, but you'll also have a section within your results to talk about it, right?
Okay.
Right, so this will be a results section too.
Okay.
Yeah, exactly. Results will be longer. Discussion should be longer.
you haven't done anything?
Methods will be longer.
Dude, at least this is the
Intro
only
should
thing I have to
actually intro
do.
doesn't need to be
Just
that
I'll
add
say
it.
that.
It's gonna take
Intro's
me
mostly
like
background, right? So you talk
couple
about your
hours, maybe.
disease and your gene also.
very
So
big
probably the
one.
conclusion of your intro will be a little longer, because then you can talk about we also
Have
did
you
all
done
these
this
You
other
got
things.
so much to do.
Wait, have you done this part?
Paper
or-
to do.
And then,
Oh.
well, if you've done all the figures already for most of the things, it shouldn't be. There'll be a lot of writing, I guess, for methods, results, and whatever. But you should already have the figures, or you could have taken Thanksgiving break
I mean,
off and just
the
show.
pictures.
Thanksgiving.
Okay.
Thanksgiving was Thanksgiving.
So this is one of your figures.
Thanksgiving, bro.
The next thing I'll do is the protein structure. So if you want me to do theirs,
Singe. Damn.
Singe, because I
Oh,
haven't done Singe. Uniprops. So first thing you want to do is go to Uniprops. And I don't really care which protein you put, so you can put the human homolog protein, so for you FSCN1 or 2, or you can put both, or you can put the fly, like whatever you want to put, or you can put the fly version, which would be Singe, right? So let's do Singe first. So the first, once you type in Singe, it comes up as Drosophila melanogaster, protein Singe. Click the entry on the left. So let me show you that again on here, entry. And once you get to the page, just kind of scroll through, and you should see a structure here. Kind of pick the best pose that shows the most like differences, and then this is it. Just screenshot this into your paper and just talk about the protein, right? There's a couple things you can talk about, like it seems like there's 2 major globular sections or
that's
whatever,
pretty
right?
easy.
This could potentially be a beta barrel actually. That's kind of interesting.
You're supposed to also talk about the structure of the protein.
Just scroll through the page.
You're supposed to tell you everything.
Yeah. So
This is something you put this in like the introduction, right? Or like only a figure?
No, this is part of the
Figures.
protein blast and all that stuff. So that would be part of your results. You can stick this with protein blast if you want, because it's all like protein stuff. But all results.
Okay.
So everything pretty much results.
Okay.
And
So
then
that's
I guess
for
the
bonus
methods
points
from, like,
for the figure,
the
but
detail
just
stuff.
if you're gonna put it in, like write about it obviously.
I guess there's no intro.
And then
I have
since
to add
we're
the
talking
gel.
about FSCN1,
So that's the methods I have to change.
You
for
said
gel.
I took
FSCN2,
pictures
right?
of,
Yes.
of the paper that we
That's the
had.
one related to the.
Ramon, text me or
Yeah, the
remind me so I
blindness
don't
one.
forget.
Or,
Maybe
okay.
start in
I'll
Yeah, because the
post
other
this
ones
on
don't really have a
Okay.
disease.
Just remind me to send the
Yeah.
pictures
So
of
here's
the
FSCN2.
the manual.
So actually, if you do a side by side of FSCN2 as well as Singe,
Okay.
there's actually
No,
2
look.
general globular structures that is kind of similar, because this is FSCN2.
Are we supposed to compare it?
If you want, there's just more stuff to put there. It always looks better, right?
Okay.
The more, the better. Alright.
I like that plan. That's a good plan.
Yeah.
So,
2 globular areas.
okay. So, that's it. There's your figure for bonus points, 15 bonus points.
For real?
Where?
Yeah, so if you look
Swear?
at the
Don't play.
Don't play with me.
No, if you look at the thingy, the rubric,
Uh-huh.
apparently it's for bonus points. I don't know. In the past, we always required it.
I'll get it done
Oh,
tonight. What
dude,
you mean?
this guy.
I'll do this when I get home.
Praise the Lord.
Bro, you got your,
This
you got your,
figure?
What else? So there's this. I mean, it might get changed. I don't know, but so far, currently in the rubric, it's for 15 points.
it should be with reservation. You have to do
Right,
the rest
and
of
then
the
protein blast, you guys
assignment.
have done. I just did it. Okay, so I guess the final thing is discussing all the figures that you'll actually need for that paper.
Okay, that's actually
That's actually what the question I was gonna ask.
Yeah, just make sure you talk about it. Yeah, that's
Because
I mean,
honest
you can
points
do
on
side by
pictures.
side with wild
Yeah.
type because you don't have to.
yeah.
whatever you want.
That's why I was like.
No, you weren't in. I was saying.
So it
Oh,
could be,
wait-
it could
wait
be for
to-
either
wait to
the
group.
flies or for the human homolog.
Wait,
Camera
Matilda,
shy.
did you have all the figures?
I got a few points off.
All right,
I don't know.
so let's start with the
Have all of
figures.
them?
So you guys know about the gel figures, yes?
Gel?
Right, the you need to crop the gel and then do all that stuff for the gel. We still haven't given you the ladder sizes, so we will do that. There will be an amplicon. You know what the amplicon is, right? So pretty much the Word document with the 2. Other than you guys, you'll just have 1 primer. For everybody else, you have 2 primers, and the sequence in
Wait,
between.
for us, what
Yeah.
one?
Mhm.
Okay.
Two.
Wait, two.
Two.
I was paying attention to these
You
points.
said one for someone else.
Okay, so that's your amplicon figure.
Gel and
The next thing that you will show, I guess, would be your nucleotide blast. And the blast that really is part of like all this stuff. But so when you do your alignment, like the second tab is like actually I probably should have showed you guys, but I'll show you after this. But pretty much it's a page with all the alignments, right? And remember I told you to go to the individual differences and like pick it out and find the peaks and put them together, right? Just the nucleotide blast is just the whole thing. So you crop the whole thing out and just paste it. If you guys have a deletion, so you guys have deletion, it'll be 2 sections, right? It'll be like one section and then a second section. So just include 2 sections. You guys will see 2 sections also. Just include those 2. You guys will just include 1 giant section. You can do 2 of them for you guys since you're looking from both sides for mutations, right? And vestigial, I guess you can, when you do it against, you guys do wild type singed against your sequencing data, right? Yeah, so either way, just find a piece of nucleotide blast and just include it. That's not a big deal, but just include some. And you know what it looks like. It looks like this, right? Like your top is your whatever, your query sequence, your subject, and then all the vertical lines that show the alignment. But then some of them will be like a dash or like or whatever, which means it's not there. Yeah. Okay, so just include that figure. I'll show you what it looks like in a sec. Okay, so that's your nucleotide blast. After your nucleotide blast, you'll include all those things I talked about earlier. So like U2 will be dot plots that like they look like this, right? Remember this thing with a little gap and on the line?
Mm-hmm.
You'll just show this because that's your major figure. So make sure you have that for you guys. Make sure you have this like multiple of this figure, which are your SNPs and indels. Because for you guys, that's your mutation. Vestigial, video 4. Yeah, video 4 for vestigial, and that's it, right? But everybody, if you can, it always doesn't hurt. It'll only help is if you do, so for black, you will only have this, but for everybody else, if you can find SNPs and indels, it's only gonna help your grade, just saying. So it doesn't hurt to include both your dot plot as well as the SNPs and indels. You guys remember what the SNPs and indels are, right? Like you pretty much go to your blast data, you find the pieces that aren't aligning, and then you go back into your Finch data to see if the peaks look good. And if the peaks look good and you have the mismatch, that means it's a potential indel or deletion or whatever, right? Yes, no? Yes, okay. If not, I can show you guys. No.
Safe to show us.
Yeah, no, yeah, exactly. So for black, it doesn't matter as much because it's, right, it's, there's no like giant shifts or whatever that you can see. So it's not that big of a deal.
I need a bed. That's all I need.
Okay, okay, so that's not done yet. I mean, I'll send an email out about this, but then it's the 2 figures I just went over, which is the protein blast, and then after that the crystal structure, which is the UniProt, right? Whatever I pulled from UniProt, that 3D structure. So 3D structure, protein blast. Cool. So those are all your figures that you need to include and write about. I mean, like the things to write about are E values, S values. Differences that you see, so parts that are not aligning, yeah, and yeah, just talk about it, right? Just, hey, we see these things, whatever. Okay. Is there anything else we need to discuss? Any questions about anything?
Are there videos for everything?
Yeah. Yeah. So for the protein blast, that's end of video 2, so you kind of have to skip through half of it. But pretty much, yeah. So just video 2 for protein blast. UniProt crystal structure. I think I quickly go over that in like video 5 or something. Even though like it'll seem like I never talk about it because I talk about another gene which is yellow. But if you skip through it, you will see it. But pretty much just Google UniProt and Google your gene and find a picture.
Just
Right,
watch
it's-
every video of yours.
And then yeah,
Just watch his videos and
that
like,
should be it.
didn't help at
Okay, so
all.
if you've turned in your
Did you
sub,
watch
oh
it?
yes,
I haven't
does somebody
watched
have a
The-
piece of paper?
The video too.
Oh.
We'll pass
W2.
around, and then I'll
Or
steal a
W-
pen
yeah,
or
or
pencil.
W2.
And then what I want you guys to do is write your name and PSID, and that will be for your 10 bonus points. So as I promised, that if you showed up to class, I would take roll one of these random days as a spot check. I never did it, so today's the day. So if
The
you
last
showed up
day.
the last day of
The
class-
last day.
This
I
would
was
be
this-
the-
I was this close to not coming too.
Bro, the way you're talking today, I was thinking you may not have come. And where I didn't see you walk.
Yeah, when I was late.
Okay, I think it's it.